Well, good morning and welcome to this parallel session on the subject of how did life begin, and what a great question. And I do believe this is one of the greatest questions that science tries to answer. One of the greatest questions possibly of all time. And let's start with some known facts that are irrefutable, and that's all life as we know it is coded by DNA. Perhaps the one exception, whether you can call it life, is something like a virus that's got RNA, but it still uses the same code system, the codon. And so all life is coded by DNA. Secondly, all life as we know it comes from life itself. All life as we know it comes from life itself. So the question is that we're trying to answer, how did the first living cell, and this is what evolutionists would call the first living cell, arise? Now that living cell needed to be able to replicate itself in some way or another, to contain that information that is built up over time according to evolution. But how did the first living cell? And one of the things that I would like to put to you this morning is that even the simplest cell is amazingly complex. In fact, one of the simplest life forms that we have in existence today in the natural world has at least 365 genes that are required to make the proteins, the enzymes, and everything else for that cell to replicate. And we'll come on to that. So does evolution, which is a theory, have a scientific answer to the origins of life? In fact, this was my own personal question back in 1984. I thought this, that if I could in some way find out how life began, I might know what life is all about. So what is life? And that was my big question. And all my teachers at that time, when I was studying my A-levels, said that I should study biochemistry. And so I picked up this little book in Blackwell's bookshop in Oxford, where I lived, called The Chemistry of Life by Professor Stephen Rose. And what I discovered in that book, before I went to university, that life was much more complex than I had previously imagined. I was very much influenced by a series that ran at the end of the 1970s called Life on Earth. And I'm sure you recognise this great presenter, Sir David Attenborough. And I was strongly, as a teenager, influenced by that series of programmes. And in particular, an episode where he spoke about the layers of rock in the Grand Canyon. And he said that each layer in the rock strata represents 1,000,000 years. And so as you go deeper down into the canyon, which has been eroded by the river that runs through it, you're going back in time and you see the simpler forms of life in the fossil record. And that was something that really caught my attention. And I wanted to know from that point the detail of how life actually began at the very start. Well, it's only a matter of time, I thought, when I went to the University College of Swansea in South Wales to study biochemistry, that I would have the answer to how life began. But none of my lecturers, and believe me, I asked every one of them, could give me an adequate, reasonable answer to how life began. They could not answer the question. And I was surprised, because the way that David Attenborough presents is that this is not a theory, but a proven fact. And therefore, if it's a proven fact, we must have all the evidence. But they had no evidence that they could offer. And there were other questions that arose during my first year at university studying biochemistry, particularly the whole matter, which I'll speak of very briefly, on protein synthesis. And how could such a complex machinery that's involved have evolved by chance? So these were some of the questions that I had towards the end of the 1984. And then this was the final straw, really. This is chapter 32 of a textbook called Biochemistry by Jeffrey Zube. And it looks at the origin of life. And so I devoured that particular chapter. And you can see on the picture to the side that of a comet. And the reason being is because they could not give a definitive answer how life began on Earth. And so the suggestion is that life may have started elsewhere in our solar system, and this Earth was seeded with life or with some of the important biochemical molecules, the organic molecules, such as cytosine and adenine and the things for DNA, and phosphorus as well. And so all of these things perhaps were seeded by comets entering our atmosphere, melting, and the meltwater containing these organic molecules collected in the oceans. And so this was, as I say, the final straw that led me to reject, on the basis of the lack of scientific evidence, the theory of evolution. And I was, by all admission, lost and didn't know where to go, until I went to a Christian Union carol service. This happened in my second year at university. And on the 10th of December, 1985, I heard about the reason Jesus came into this world. And that was to die for a number of people, to save them from their sins. And I knew by that time, by my lifestyle, that I was, by all accounts, a guilty sinner. And I needed a saviour. And I did a very traditional thing. I went back to my university room and I knelt beside my bed and I spoke to God and I said, I don't understand everything, but what I do know is I am a sinner and Jesus is the friend and saviour of sinners. And I asked him to save me. That changed my life entirely, including my worldview and the answer to this question, because now everything made sense. The illustration I sometimes use is I was trying to put my foot with its 5 digits, my toes, into a glove. And though I might try to put that glove upon my foot, it didn't quite fit. But when you put your hand into a glove, then you can see how it was designed. And so the theory of the natural world, as we see it, and creation made sense. I began to see that this world is designed. And the precision engineering, and you would have seen some of that bacteria flagellum, which is 99.9% efficient, a simple microbe, but it's so small you can't see it, even with the most powerful light microscope. And yet we now know that that is required, all those various protein parts for it to work. So there is a design. So I was convinced of the design element. And as I say, creation fitted the complexity of life like hand in a glove. And so I'd like to first of all speak about the origin of life as being what I would consider the blind spot of evolution. In fact, some evolutionists say that the chemical evolution or abiogenesis isn't part of their theory at all. They have to start with life, and then they explain how it moves forward. But you need to understand, how did that life first arise? It is a question that they are avoiding because the science is so strongly opposed to abiogenesis. So it cannot explain, for example, how molecules organize themselves in the way that they do in the natural world. It cannot explain how cells, with all their internal complexity, arose. And it cannot explain how the very first cell was able to replicate itself. Or in other words, there was no biochemistry, no cells, and no reproduction. And these are pressing questions that anyone looking at the origin of life. Neither Darwin nor his many followers can tell us how life could have come from non-life. Now, just because something is organic doesn't mean that it is living. So methane, CH4, is an organic molecule, but it doesn't mean it's a living molecule. So that's important to understand as well, because you're talking about organic chemicals coming together in high concentrations. And I would add, which I haven't got in these slides, that many of the component parts that are necessary for life to exist are very difficult to manufacture, even from a synthetic chemist's point of view. And they do not occur naturally, and certainly not in the concentrations required. They are very unstable. And particularly if we're talking about the origin of life happening 3.5 billion years ago, as evolution suggests, the Earth was just dipping below 100 degrees Celsius. So not only do you have the problem of this warm pool, but it's a very hot environment for life to exist in. We'll look at the environment where life supposedly existed, which is actually life couldn't exist today in that environment because of particularly the ultraviolet radiation. We'll come on to that in time. So let's follow what I call the fairy tale of how life supposedly began, not just millions of years, but billions of years ago. And it's commonly taught that the earth formed, we heard it in the talk earlier, that many secular scientists believe and teach that the earth formed 4.5 billion years ago. It's a bit like once upon a time. There's no evidence. It's a story. And it's something that is taught to children in primary and secondary schools. And children, rather than being educated and taught how to think for themselves, are indoctrinated in a particular secular, atheistic, materialistic theory. And then the earth supposedly cooled, sufficient for life to arise. But there's something else as well, that there is a need for water. Where does the water come from that is so necessary for life? It's believed that the comets supplied the waters and they filled the oceans. What they don't tell you, because they haven't got an alternative explanation, is that water has a fingerprint. What do I mean by that? Well, there's a certain stable isotope called deuterium. And deuterium, or heavy water, has an extra neutron in the hydrogen, and therefore it makes it slightly different as a stable isotope. Well, they've now analysed the water from comets, and they have found that the proportion of deuterium to hydrogen is very different to what it is in the earth. So it does really, forgive the pun, blow that whole evidence out of the water. That we now have the scientific evidence that comets didn't supply. But this is the theory, and we're following the theory of evolution. And so at this time, the atmosphere also didn't have oxygen. Now we know that certain life forms, anaerobic bacteria, for example, don't need oxygen, and oxygen can actually be quite toxic. So it didn't have any oxygen. That oxygen came about through the early part of the evolution of plants and cyanobacteria, which you will often get told. And the chemicals began to, this is the story, the chemicals began to form and accumulate in the sea, including organic molecules like amino acids. Then, in one of these pools of water, and we're going to read a letter that Charles Darwin wrote to a friend in 1887. If, and oh, what a big if, we could conceive in some small, warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed, ready to undergo still more complex changes. At the present day, such matter would be instantly devoured or absorbed, which would have not been the case before living creatures were formed. Well, amino acids, let me tell you, do not spontaneously bind together without enzymes. And let me explain why they don't bind together. This is what we call a peptide bond. You've got two amino acids here, and on one side of an amino acid you have a carboxyl group, COOH. So you've got that OH group, and you've got the amine group on the other side of another amino acid, and one of the hydrogens of those bind together or let's say join together with the hydroxy groups to form this peptide bond. And this is called a condensation reaction. So water, as you can see in that tiny molecule at the bottom, is emitted. And so there is the joining of the hydroxy and amino group, and then water is formed. So water is, now I want you to understand this, water is removed. And we know that many organic chemical reactions are a two-way street. They are in equilibrium. And if you take out water, then you drive the reaction forward. But if you put water in, it drives it the other way. So otherwise, in an aqueous environment, the water that is in the medium actually causes amino acids to dissociate faster than they associate. So amino acids do not bind together to form peptides without some form of energy. Water is not favourable for peptide bond formation. So I don't need to talk about the deep ocean vents in the bottom of the ocean, because they still contain water as well. So to work against this dissociation, you need energy. And not just any kind of energy like sunlight energy, which is random. It's like putting a bull in a china shop. Lots of energy, but no order resulting from it. But you also need something called enzymes. And every biochemist knows that for a chemical reaction to take place, you need an enzyme, which is a protein molecule, to bring the two parts together or to cut them apart. And of course, enzymes are proteins. So for proteins to form, you need a whole set of proteins. So where did the proteins originally come from? That's one of the important questions. Proteins are themselves, and this is something that if you're not a biochemist or you haven't studied biology at A level, you may not be aware, but there are molecular machines within every eukaryotic cell. So we have ribosomes inside, and we're going to look a little bit about ribosomes. I know my son Jacob, who's also a biochemist, spoke a little bit about them yesterday. But these ribosomes, these very complex machines themselves are made of RNA and protein. But let's go back to an experiment that tried to replicate or mimic the early Earth. And two scientists, Miller and Urey, set up an experiment that aimed to mimic the early Earth. They used, for example, without the oxygen being present, a reducing atmosphere. They contained certain things like methane and ammonia and a certain amount of spark to simulate the lightning that would have coursed through the atmosphere at that time as well. It's a very famous experiment. And there are some conclusions, but before we come to the conclusion, there is an assumption. And the assumption is that the early Earth had itself a reducing atmosphere. In other words, there was no oxygen. And the problem is, if you have no oxygen, which is O2, then you have no ozone, which is O3. And ozone goes up into the stratosphere and it forms a protective layer which filters out 95 to 98% of the harmful ultraviolet radiation, which certainly would have zapped any early form of life that existed at that time. But the products also are very interesting. There was an organic soup containing a few amino acids. This experiment I liken to the SETI program, the search for life. And they only found only a handful of the 20 plus amino acids that are required for proteins. But not only that, not only were there only a few of the proteins, and they certainly didn't contain many of the nucleotides, which we have time to go into this morning, but the problem was that the amino acids were the wrong kind of amino acids to form proteins. What do I mean by this? Well, as any chemist knows, that amino acids form in a natural way, if you find amino acids, in a 50-50 ratio. They are isomers of one another, mirror images. But only the left-hand form is used in proteins, not the right-hand form. And likewise, with the nucleotides, only the right-hand form of nucleotides form and not the left-hand form. So it's a big problem when it comes to...
trouble with the connection. Please try again in a moment.
There's a real problem when it comes to form the proteins. And let me explain why. Number one is this, that just 1 to 2% of the right-handed form of amino acids is sufficient enough to cause proteins to break apart. So if you've got too much, even as little as 1% of a right-handed protein, it would break apart and wouldn't form a stable protein. Secondly, as we've already seen, peptide bonds involve the removal of water, so that negates against protein formation. And also, as we will go on to speak about now, the biological proteins are made by these very complex molecular machines. So let's look at this one in particular. And this is a spatial model of a ribosome unit. It contains two parts that come together a little bit, as Jacob said yesterday, and I'm going to repeat what he said, a little bit like a tape recorder. So these machines are only from one end to the other, at its widest part, 20 nanometers across, which means that you can get 50,000 of these lined up side by side would make up 1 single millimeter. And yet these are very precision made machines, biological machines, that are necessary. So even something like a virus, like the coronavirus, it will inject into the cell the RNA, and then through reverse transcription you can get the DNA, and then that will replicate itself and form many more RNAs which run through the ribosome. So what a virus does is hijack the molecular machinery, because even a virus cannot replicate itself without this machinery. And I can't think of anything, if you call a virus alive, and I have my doubts on that, whether even the simplest form can actually replicate itself without a ribosome being present. So ribosomes decode a message. We heard about this thing. You can see on this thing a codon. It's made up of 3 nucleotides, say A, T, and G, for example. And this would correspond to like the thread of information that runs through the ribosome unit. And amino acids are brought in by what's called transfer RNA. I look like little taxis. And each transfer RNA has a codon counterpart at the base, and it has an amino acid binding site for a particular amino acid on the other. So as it goes through, only that particular tRNA will come in, bring in the amino acid, and so you get this protein. And it's not like something like starch, which is just made up of a chain of glucose molecules, but it contains information, because the amino acid sequence is essential for the protein structure and its functionality. So without that protein structure, it's a bit like you've got 26 letters of the alphabet and you've got thousands, if not millions, of words, but all of them made up. But for a word to make sense, the letters have to be in the right order. So for biochemistry, it must start with a B and then be an I and then an O. So there is information coded within this messenger RNA that moves through the ribosome. And so the ribosome is a little bit like a tape recorder itself. And here is a tape recorder. You've got certain parts to this. You might have not seen one of these. I used to play about with this when I was a little boy, before even the compact cassette came about. And now, of course, nobody knows what those are. We talk about CDs. Well, what are they? Well, they're an early form of MP3, and we'll stop it there. But technology moves on very fast. But here is a machine, and it has certain parts, particularly the messenger RNA. It's like the tape. And that magnetic tape, it contains the information, but it needs to be read. You don't look at magnetic tape and say, what a great song. You have to play it through the tape head, which decodes, as it were, the magnetic part of the tape and turns it through voltage fluctuations into sound and music. And so a tape recorder has those particular parts. The tape is the all-important thing. As it passes through the tape head, the information is read and processed. So ribosomes themselves, 30 millionths of a millimetre across, and yet they are as complex as a tape recorder. But here's the other thing. A tape recorder requires some form of energy. We're not talking about something you wind up, but something you plug into the electrical socket. You need energy. And all ribosomes, for every single amino acid that is added in a sequence in a ribosome, requires a form of energy in the form of ATP, adenosine triphosphate. And it doesn't matter whether you are a bacterium or a plant or a virus, well, maybe not a virus, or an animal, they all run on ATP. Or it might be GTP or something like this. But ATP needs to be present. And we saw in Jacob's talk yesterday how ATP is synthesized in that electron chain, transport chain, in the membrane of mitochondria in eukaryotic cells. But in prokaryotic cells, it still uses ATP synthase to form the various amounts of ATP. So this is part of the energy. So glucose, if I had a teaspoon of glucose and set light to it, there would be an explosion. It contains so much energy. So this is a controlled explosion, a little bit like a combustion engine. It's broken down in parts, small parts at a time, and every glucose molecule makes about 36 ATP molecules. And this needs to be present, let me say, in the first primitive cell. So you need the ribosomes, and you also need the form of energy through which is the end point of metabolism, whatever that source may be. And so what we put forward is that science is the greatest enemy of evolution. It refutes evolution. This is an excellent book that you can get, I believe, from CMI. Might be available on the bookstore. And here is another book, The Biochemical Challenge to Evolution, Darwin's Black Box by Michael Behe, who talks particularly, and he doesn't come as a Christian, but he comes as a scientist looking at things, and he could not have enough faith to believe that these things came about by chance. So the complex world of biochemistry and molecular biology cannot give an answer to our question: how did life begin? So let's look at something else. First of all, this earth, which all life in our universe found to date, the only place where life has ever found to exist is on planet Earth. You go to the moon, it is sterile. There is no life. There is no life yet found on Mars, and I don't believe they will ever find life on Mars. Earth is the only planet with life. Oh, of course, Earth has 71% water coverage, which also regulates the temperature as well as other things, providing the water that we need for life. And of course, we know that water is essential to all living things. So come back to my original textbook. Did life come, if it didn't arrive on the earth, because they cannot explain how it did so, could life have come from outer space, perhaps seeded by comets or meteorites? The recipe for life comes down to this, according to this very simple idea. If you've got water and you find amino acids and a few minerals, hey presto, you're bound to have life. And the only reason we know that life must have started in this way is because it exists today. So that's the very simple thing. So did Mars, for example, once have water and therefore harboured life? This has been a question particularly in the last century, but also the present one. So here are some mock-up pictures of what Mars may have looked like about the same time that life started on Earth. Could it have started on Mars? But we have sent probes to Mars, and though they have dug right down beneath the surface, they have not found any clear, hard evidence that life ever existed on Mars. There was a fossil put in the papers, I think in the late 80s, well, probably the mid-90s, when NASA was beginning to deplete in funds, and they said, we have discovered life on Mars. We have found a fossil of a bacterium on Mars. And yet when they examined it more closely, they thought, well, we might have to think again. It might just be a mineral deposit that looks a little bit like a bacteria. But they got the funding to continue their exploration. So it was a little bit of a propaganda And so you do need to be careful what you hear about great discoveries. Is there life, for example, under the icy crust of Europa? And there is a probe that is planned to land on one of the moons that goes around Jupiter. And they believe that under the icy crust of Europa, there could be a warm lake underneath, and life will be on there. And also Enceladus, which I haven't got time to go into. There is another exoplanets. They're finding if there's water on exoplanets and it's the right temperature, then it must be their life there. So this planet called B218b, I'm sure you've heard of it, only 110 light years away, which is a bit of a problem because it would take us, with our known technology, 1 million years to get there. And then you've got this other probe, Philae, which came from this probe that went round a comet. And it sent this washing machine-sized probe. It bounced on the comet, and it bounced into a shadow. And so the solar panels didn't really operate. But as it came in towards the sun, they came back to life. They looked for signs of life, but no life or the right molecules were found. So the thought remains that comets have seeded the Earth with the essential molecules. So you can't get more cutting edge than NASA, but they cannot give an answer to our question, how did life begin? And so, in conclusion, the great enemy of science is the fairy tale of evolution. And many people are self-appointed experts in origins. This pseudoscience describes how man arose from a fish, and a fish arose from something much simpler as well. We're not getting into that at the moment. But here, you might want to meet one of your ancestors. I don't know what your family tree looks like, but this looks something a little bit alien. Does anyone know what it is? It's a mud skipper. And the idea that they came out of the water and were able to breathe some of the oxygen and find their way onto the land. Or let me look at this question. Answer to the question, what came first, the chicken or the egg? Well, the answer that scientists give, evolutionists give, is an amoeba, some kind of creature like this. But we've got a similar question. Which came first? The protein that is so important to make the DNA, or the DNA which codes for the protein? It's the chicken and egg situation, but in a much more scientific and fundamental level. And this whole area where we've looked at the translation of messenger RNA, the code, into proteins, has won many Nobel Prizes, particularly here in the 1960s. So we know that DNA codes for proteins, and that proteins are needed to facilitate both transcription and translation into proteins. But I want to just bring your attention to one particular protein we looked at yesterday. The complexity doesn't stop there, because proteins, there are things called motor proteins. And these, like this particular kinesin, they're folded into a defined shape. They use energy in the form of ATP, and they're able to walk along that microtubule. So here's the question. DNA is like the software. Proteins and ribosomes are like the hardware. So any computer engineer will know that if you've got the greatest computer, if it doesn't have any software to operate on it, it's useless. And if you've got software without the hardware, again, it's useless. You need both together for them to have any functionality at all. So DNA is the software, protein is the hardware. Both are needed at the same time. And so we now know that complex machines, to work, they need both designers and engineers. So the creation model takes that deduction that we need a designer and an engineer. How much more for the biological machines that are so essential to life? Life can only come from life. And so here is the evolutionary model. From some kind of rock or organic soup came life. We know that that cannot be the case. We know that life comes from life. Here's a simple case of mitosis. And even the simplest life requires, in fact it's slightly lower, 265 genes to code for the hundreds of proteins. And so there is not a single element of hard scientific evidence that can account for life having started by mere naturalistic means. And 164 years on, since Darwin wrote his infamous novel, there has been no scientific account that can account for how life started. That is a scientifically proven fact. So when you see precision engineering, is it not reasonable? God has given you a reasonable mind to deduce that creation is by design. Organisations like Truth in Science find that science and faith fit together. And our argument is from evidence-based science while we still have saving faith. And so I would put to you that life, rather than evolved by some fairy tale in a galaxy far, far away, actually was created by God here on earth, just as is recorded in the Bible. Thank you for your time, and I'll take a few questions before we need to move on.